Hello everyone, welcome back to Meds.Concepts. Today we are going to study an important and classic microbiology technique, Albert staining. This method is mainly used to detect metachromatic granules, also known as volutin granules, in bacteria. In this presentation, we will cover the principle, procedure, results, and clinical importance of Albert staining. Let's get started. Albert staining is a specialized differential staining technique used in microbiology. It is mainly used for identifying metachromatic granules inside bacterial cells. These granules are made of inorganic polyphosphate and act as storage material. The technique is especially important for identifying Corynebacterium diphtheriae, the organism responsible for diphtheria. It also helps differentiate pathogenic bacteria from non-pathogenic diphtheroids. Now let's understand the principle behind Albert staining. This staining is based on the property of metachromasia, where certain structures stain in a different color than the dye used. The metachromatic granules take up a blue-black color, while the rest of the bacterial cell appears light green or light blue. This contrast makes the granules clearly visible under the microscope. So basically, the stain highlights specific intracellular structures rather than the whole cell. Albert staining involves two main solutions, Albert's A and Albert's B. Albert's A solution contains toluidine blue, malachite green, acetic acid, and ethanol. This acts as the primary stain. Albert's B solution contains iodine and potassium iodide, which acts as a mordant. The mordant helps in fixing the stain inside the granules, enhancing visibility. Now let's go step by step through the procedure. First, prepare a thin smear of the bacterial sample on a clean slide and allow it to air dry. Then perform heat fixation. Next, flood the slide with Albert's A solution and leave it for a few minutes. After that, add Albert's B solution without washing. Finally, rinse gently with water, air dry, and observe under the microscope. After staining, we observe the slide under a microscope. The metachromatic granules appear as blue-black dots. The bacterial cytoplasm appears greenish or light-colored. In Corynebacterium diphtheriae, these granules are usually seen at the poles, giving a Chinese letter arrangement. This characteristic appearance helps in identification. Albert staining has important clinical applications. It is used for the presumptive diagnosis of diphtheria. It helps in identifying Corynebacterium diphtheriae quickly in clinical samples. It is also useful for differentiating pathogenic bacteria from harmless diphtheroids. Additionally, it can detect granules in other medically important bacteria. Now let's look at some limitations and precautions. This technique is only useful for bacteria that produce volutin granules. Fresh cultures are important, usually 18 to 24 hours old, because older cultures may lose granules. Also, proper biosafety precautions must be followed, as Corynebacterium diphtheriae is a pathogenic organism. To conclude, Albert staining is a simple, rapid, and effective diagnostic technique. It plays a key role in identifying Corynebacterium diphtheriae. The ability to detect metachromatic granules makes it a valuable tool in microbiology. Even today, it remains relevant in routine laboratory diagnosis. So that's all for today's video. If you found this explanation helpful, make sure to like, share, and subscribe to Meds.Concepts. Don't forget to press the bell icon so you never miss upcoming microbiology concepts. Let me know in the comments which topic you want next. Keep learning, stay consistent, see you in the next video.